So, welcome to my sermon. I'm very happy to be here, in particular because if I would be in Germany now, I would be like this now. Because in half an hour the semi-final and the Champions League between Bayern München and Real Madrid will start, and I'm extremely nervous. So, if you know some goals, don't tell me, it will destroy me. So I will speak about the first half of my presentation is more on the conceptual method side, and the second half I will have an example. So a brief overview, type of models, and then we speak in this workshop about predictive systems models. So prediction is a key term, and I think it deserves some attention. The role of multiple patterns to make the models predictive and test them. The very important role of really distinguishing emergence from other ways of getting your models look good. It's the individual stupid, which is the American style phrase of emphasizing that individuals are really important in this entire business. I think there's a broad agreement here. And we haven't touched so much so far on communities and ecosystems. So I have one example model, a plant community model, but it is a demonstration of general principles and approaches which I think should be useful for extrapolating from perhaps molecules, but at least from individuals to communities. I will not so much speak about ecosystems. So the types of model, Valerie mentioned them yesterday. I will not go into detail here regarding terminology. Roger, now I'm the master. But by the way, individual-based was a term which was used a lot in ecology. Agent-based was a term used a lot in social sciences. There have been differences. Ecologists have been focusing on differences among individuals, on local interactions. This was often the most important motivation to use this kind of simulation models. Whereas in social sciences, people have often focused on decision-making, adaptive behavior, so behavioral ecologists. So there were historical differences, but these fields are now overlapping more and more. So I think it doesn't make any sense to distinguish these very much. So I was strongly fighting with people using ODEs and matrix models 20 years ago. Maybe I'm getting old, but now I think, oh, they have all their pros and cons. I'm getting mild. I love you all. It is true that they have definitely their pros and cons. And also the differences have become less strict than they used to be 20 years ago or 40 years ago, because most models, independent of what type it is, are now run on computers. Even if you say a matrix model or an ODE model, all the models which we have seen in this morning, they all run on computers. You could argue, is it a simulation model? Is it a mathematical model? It often very much boils down on how you formulate your model. Just use equations or a set of algorithms. And actually, most individual-based models are hybrids. So, of different model types. For example, the population models of Daphnia developed by Ben, which Roger has mentioned. The Daphnia individuals are described by the depth equations, so differential equations. So this long debates about matrix and structured and IBMs, I don't go there anymore. I have to say that ecology has for a long time favored simple models over complex models. Theoretical ecology, theoretical population ecology especially, was focusing on general concepts and understanding. And prediction was considered something which is not so important. First, we need to understand everything. This seems to be changing now. So Matthew Evans, who is sitting here, has been very active in the last year, organizing 2 workshops and leading 2 papers. By the way, whenever Matthew invites you to come to a workshop, accepts the invitation, he will make sure that the paper will appear. The same is true for Valerie, I have to say. So it is now possible to publish such a paper in Transient Ecology and Evolution. It would have been impossible 10 or 20 years ago. So the influence of Robert May is slowly going away. I'm not saying he's a bad guy, but he has defined a certain type of models, and I think we really need a diversity. There's an entire special issue in the Philosophical Transaction of the Royal Society, edited by Matthew and his colleagues, on predictive systems ecology. And so also Valerie and Peter have referred to predictive systems models. So this is kind of People were talking about this in the 60s and 70s, systems models, but this was a completely different style. And ecological modeling and system theory has made many, many, many achievements. Individual-based modeling, one part of it. So the situation is changing. But a key problem is that in ecology, the term prediction has been kind of diluted or is not used in a proper way. In ecology, any model output is called prediction. You very often have a paper that at the end, you have here some variable, often it's time or whatever. You have some output variable, and then you have your observed values, maybe some replicates, and then you have some output of the model with some confidence interval. So this is called here prediction. Yeah? Model output is called prediction. And very often it is not. There's no culture yet of communicating to what degree you have calibrated your model. As we all know, modeling always includes some tweaking and calibration. There's no way around it. But this could be just a fit, right? So you could have 10 parameters and you were completely fitting the entire parameter set. So this is nice to have and useful for certain purposes, but this is not a prediction, right? This is a fit. And this distinction very often is not made. It could also be, I had one case where people have developed a honeybee colony model and they were showing in the course of a year in the season, well, this is what the model predicts. The colony size starts in January, growing in summer, and then they have a data value here, data value here, and say, hooray! The model is a total perfect, it's very realistic, but then if you have a second figure, which I do not show, which is the egg-laying rate of the queen, which was completely imposed, right? So people often also do not communicate in a clear enough way how much your good results have been determined by one decisive environmental driver. So this culture of communication is not there. And often people, and I will detail on that later on, model data comparisons are often just based on one single pattern. You know, this typical case of there has been cycles and some small mammals or some forest birds. This is a pattern which is fancy, and then you have a model which predicts almost the same. You say, hooray, this is it. But then you find 10 other models which have exactly the same output. So how do you know now which one is the correct explanation of the mechanism? So here's the test. 2 slides now with the question to you. This is the output of a forest model and some data. So this is time, about 80 years. And we have the typical matrix which forest scientists measure in a tropical forest, the mean height of the trees, of the plot which is re-establishing after disturbance, the typical stem number, the typical basal area, so taking the area of each individual tree and summing them up, and the maximum height of the tree with the stem. And so the lines are the model output, is the standard variation, and the dots are the observed values. And we once had a visitor in our department, which was David Ford from Seattle. He wrote a very nice book about ecological method, published in 2000, Cambridge University Press, and he was talking to various PhD students in our group, also to Nadja Rieger, and he came back and he was really furious. He said, this student showed me this figure, and she claimed it was a perfect match, it was a perfect validation. But this is totally off, how can she say this? So you need to know that David Ford is a biometrician, so his perspective was more that of a statistician. So in terms of a fit, this is really, really bad. But what he did not know is that these values have been taken from data and plotted after the model predictions have been made. So there was no calibration, no tweaking involved whatsoever. And therefore we said, hooray, this is a model with 80 parameters. This took this poor PhD student a year to get the low-level parameters calibrated, not the system-level parameters, the physiological parameters. This model is a forest model driven by photosynthesis competition for light. So if you communicate how much you calibrated and how much you use environmental drivers, this is fantastic. And it is also fantastic in terms of having different metrics not only one of them. So you can see in one matrix, some aspects the model is quite good, and others it is not good. So of course it is not a perfect model. Now the next message is you could now, of course, increase the quality of the fit, right? You have enough parameters, you could do that. But this doesn't make very much sense because this is just one plot somewhere in Mexico with specific soil and climatic data. So what's the point of improving the visual fit? Now it's really about the trends and the general patterns. Another example from a recently published honeybee model where I have been involved. This is a foraging module of the model, and the gray parts are an experiment done by one of the most well-known honeybee researcher, Tom Seeley. With colleagues, he did an experiment on an island where there was no food resource except these two feeders which I provided to this one colony. One was north of the colony, one was south of the colony. And the south feeder initially had double sugar concentration in the solution than the south feeder. And the bees were quite smart. Very quickly they realized it's better to fly to the south feeder and not to fly to the north feeder. After 4 hours, the experimenters swapped these or switched these two feeders, north to south, south to north. And then the foragers very quickly realized it doesn't pay so much anymore to fly there. They recruited less to the south feeder, recruited more to the north feeder, and after some time, the preferences have been reverted. Now, when you look at the black lines, south and north feeder, you see the same trend in the output of the model. Yeah, so they realize the north one is better, then there's a switch, and then they switch to the south one. So is this a good fit or not? And I think it is kind of reassuring that they realize something is happening, but I think there are also important differences. For example, the model bees are much more strict and much more deterministic in their response than the real bees, which might be significant if you really want to use this model in a realistic context. So it captures important trends, but there are also important differences. And I think if we learn this culture of looking at model output, not looking at perfect fits, but at what are the generic mechanisms, then this entire business of developing integrated predictive systems model will improve a lot. So we should communicate, are environmental drivers involved, and how much do they really impose the output? Sometimes they do. The proportion of calibrated parameters, calibration is not a bad thing, but the more parameters you have calibrated, the less impressive it is, of course, if you have a good fit. On the other hand, if the fit is not good, always ask yourself, did they calibrate, or was it just really an emergent pattern without any calibration? And calibration can often be at the lower level, individual level, or sub-individual level, and model prediction in this case can be at the higher level. And carefully distinguish between what we now call model output verification. This is all types of comparisons of model output to data which you have known while you were developing the model. So you wanted your model to reproduce those data. We all need this kind of things we want to predict because we develop a model and we have to, from the very early stage on, we have to have some means to decide, are we going in the right direction or not? And for some complex models, like a forest model or behavioral model, it can be really something to get this right. But the gold standard is model output corroboration, often called validation or secondary prediction. So make the model predict something which you actually did not know or even think about while you developed the model. So thereby reducing the risk of tweaking the model results. There are two papers which I have uploaded to the NIMBioS website. We call it now evaluation. The idea is that the term validation is completely corrupted. You can't use it for any. If I say validation, I can go out and you can discuss it for the next 4 hours and you will not agree. And even if I specify what I mean by validation, and I'm the master for the next 30 minutes, it will not work. But if I say evaluation, this is a kind of an innocent new term. Huh? What do you mean? And then we could talk for 30 minutes what it means. But that's a presentation by itself. We make this distinction. I think this is really important also in this context here. what is verification, what is corroboration. I don't like the word corroboration, but we had to use some other word. So, physics envy has been, so physical population ecology has been accused of physics envy. We want to be as cool as physicists, so they adopted the style of mathematical models. I would call this, fake generality is a little bit too mean. There is some truth and not error in logistic equations, but in principle they are less general than they look. So instead of trying to copy this formal generality, we should rather copy the way how physicists and chemists use the word prediction. Yeah, so they really say, this is my theory, and if I apply it to this situation, this is what the theory says, and then they go out and test it. So response to new conditions and combinations of patterns observed at multiple levels and scales. Testing new experiments, physicists can do that sometimes. Now they're reaching the limit. So now the new unifying theories about elementary particles, it can be calculated that on Earth we do not have the total energy to test them anymore. So I say to physicists nowadays, welcome to the club. So we have been there for decades, so we can't test our theories. But now there's this approach of pattern-oriented modeling. So making predictions with a model and then checking the literature, the data, expert knowledge. So collecting different pieces of evidence to still test whether your model is valid or not. So what scientists do with patterns, of course, this is very, very obvious. You see some pattern in light spectrums, you see some fancy things in your measurement. Anything which is beyond random variation has obviously some systematic underlying mechanism, some reason. And we use this pattern to inversely try to identify what is the internal mechanism. Classic example is quantum mechanics. Yeah, you put some electrical energy into a gas of hydrogen or nitrogen or whatever, and then the gas is emitting some light. And the light is not just a wide spectrum, but has very distinct wavelengths. So something systematic is going on there. Then you try various theories which can also predict these kinds of wavelengths. So this is what scientists do. We develop models or theories that reproduce the patterns, and we infer from these patterns mechanisms. So this is our job. We are detectives. So in a way, we need to squeeze as much information out of these patterns as we can. This is the kind of a metaphor here. But with complex systems, how ridiculously simple is a hydrogen atom compared to an ecosystem? I think physicists would not agree. But I think, in a way, it is true. It's there forever. It doesn't change. It didn't evolve. It doesn't get babies. It doesn't have a metabolism. So even compared to an organism, an atom of hydrogen is ridiculously simple. But for complex systems, ecological systems, even cellular systems or societies, just one single pattern very often does not contain enough information to distinguish good from bad theories. So bad ecologists tended to focus, this example here, on one single pattern observed at one level of organization, sorry if it's a typo, or at one specific scale. And this is a bad culture. So behavioral ecologists just look at behavior. Those guys look at outbreaks or cycles. Community ecologists look at species numbers or abundance, rank relationships, whatever. Ecosystem guys look at flows of nutrients. So they have all the different perspectives and focus on very restricted sets of pattern. And this should remind you of this, I call it the Valery diagram, going from molecules to ecosystems. That's the same there, of course. People working on different levels of organization tend to focus on patterns on their level of organization because they know them very well, and because their theories and models are designed to reproduce these patterns. And there's a danger. If you focus too much on your own level, you might not be able to identify what's going on in the context of the entire system. So most approaches in ecology look either at behavior or metabolism or at species numbers or flows of nutrients. But we should combine these information, and we should combine information about behavior, about population dynamics, about flows of energy and nutrients and maybe about subcellular processes and get them together for this envisaged integrated systems model. So multiscopic view is a kind of a artificial word. Instead of microscope or macroscope, you have a multiscope. Take into account multiple patterns, observe the different scales and levels of organization, and try to make your model reproduce these patterns simultaneously. And pattern does not have to be something as striking as this. It could be just that your variable of interest is within certain ranges. Or you, in one specific situation, you have observed a response of your system to a disturbance. You know it recovered after 7 days. This contains very important information. By itself, it might be easy to reproduce by a simple model, but it might be hard to get 4, 5, 6, 7 patterns reproduced at the same time. So the idea is use each pattern as a filter to reject unacceptable submodels or parameterizations. It's very similar to the mindset of what people do in statistics and model selection. It's not exactly the same, but it is very, very similar. So we have called it pattern-oriented modeling, and there are a number of publications about it, and the definition is multi-criteria design, assessment, and parameterization of models of complex systems. So you use patterns observed at different scales and levels as criteria for selecting appropriate submodels. Yeah, for example, foraging or mid-choice or energy budget. You have different candidates, and you can use these patterns to identify the most appropriate ones. I think Roger referred to some examples. And of course, you can also use patterns to identify entire sets of unknown parameters. This has been used a lot. It's very, very powerful. And it has also some similarities to what people now call approximate Bayesian computing. It is the same mindset. Use multiple pieces of evidence to get your model right. So this is in the special issue edited by Matthew. I recommend really, in the context of this workshop, looking at the other papers also as the special issue. So the purpose of pattern-oriented modeling is to develop predictive systems model, in this context here for risk assessment of chemicals, that should be structurally realistic. And by this I mean they reproduce the observed patterns for the right reasons. Yeah? So they capture the internal organization across scales of real systems instead of just taking some mechanism, which is partly right and partly wrong, and then we tweak the parameters on the other submodels to make it look good on paper, but actually it doesn't capture the system very well. I mean, the Ptolemaic model of the solar system made perfect predictions of the movement of the planets, but it was just wrong. So it violated some other principles. And of course, POM is designed to optimize model complexity. I cannot go into detail here. I heard you call it sweet spot. I would call it sweet sour spot, whatever. Or other people have called it middle ground. So as a modeler, you always are between the two extremes. You should not be too simple. You should not be too complex. And these patterns might guide you to find the middle ground, which is optimal in terms of predictive capacity, but at the same time it avoids overfitting. So this is a multi-criteria assessment of a model. I've shown this figure before. Yeah? 4 different metrics, and it gives a more complete picture than just one metric. So emergence related to the problem of what is called prediction or not, the question of emergence is very, very important. And I emphasize this because it took me several years to fully understand it, because my background is classical theoretical population ecology, where people think in terms of birth and death rates. Nothing wrong with that. And then my friend Steve Waitsberg came, and he was focusing on behavior and more adaptive decision making of animals, and he looked at our models, and he saw this one parameter, mortality, 0.6. He asked me, so what does that mean? I said, well, I have a random number for every individual. If it's below 0.6, individuals will die. So that's it. Then Steve says, but fish don't die of random reasons. I said, I know, but I don't care. One day he showed me a photo of a koi, this Japanese carps in Japan, which was extremely valuable, who had a heart attack but was reanimated by the massage of his heart. So you see in this email he wrote, see, fish don't die of random reasons. So the point is, this 0.6, where does it come from? It comes from a field study, perhaps a mark recapture study. You marked some animals, captured them again, and then you realize about 40% have died last year. Now you can use this information, you have to use this information in your model, but of course it is valid only for the conditions under which this data has been taken. I think this is well known to most of you. Whereas if you want to use a model to predict what is happening to this fish population if the flow regime changes completely, or if water is taken for cooling a power plant or the temperature is increasing, or I have an alien predator invading into the stream, then this 0.6 isn't of any value, because then mortality is actually emerging from what the individuals are doing. How they select habitat, how they interact with predators, how they hide, how they forage. And if predictions for a wide range of unknown or not yet known environmental conditions is in the focus of your model, then you have to let key behaviors emerge from lower level mechanisms. And this is an animation for this. So classical population ecology, logistic equation, just one parameter, or structured models, age or stage-specific birth and death rate. But this is, of course, imposed, empirical, and we have to use it in all our models to some degree. This is for sure. But some models are completely using it, and then we have to be aware of the limitations. Or you base it on adaptive behavior. Could be dynamic energy budget theory. It could be a fitness-seeking model where individual organisms in your model try to optimize some fitness function. And if you can parameterize this submodel for a wide range of environmental conditions, which is the case for a dynamic energy budget model, it is not restricted to a specific environment, then you can use the entire model and have population growth rate as emergent property. It's no longer imposed. And I'm emphasizing this because in my home field, population ecology, many models are still not aware of this difference. So the good thing is once we have such a model, we can use it a lot, predictive, and we could possibly use it for ecological risk assessment a lot. The bad news is developing such a model takes a lot of time. The very predictive and useful model developed by Steve Railsbeck about streamfish, trout mainly, took them 10 person-years to develop it. There's a very, very predictive model of shorebirds in England, Richard Stillman, took them 10 years to develop it. The forest model, tropical forest model I mentioned, developed by my colleague Andreas Huth, again about 10 person-years. So it goes very much beyond the time frame of an individual PhD project. This is kind of big science. Yeah, so you have to make this cost-benefit analysis, and you need to know what you want. But if we talk here about integrated systems model, predictive integrated systems model, this is not going to happen within 2 years. It takes some time. Once you have it, you can harvest for the rest of your career. Yeah? So my colleague Andreas Huth, he stopped developing models 10 years ago. He has cohorts of PhD students running through his lab and applying his model in Mexico, in Africa, in Asia, and everywhere. So this is really good. But you have to have what we say in German, a long breath for that. So patience. So the Emergence dream, of course, is, I was dreaming of this for many years, almost 2 decades. What if I have an individual and I describe it in terms of some rules, and then everything else I can see at the population level emerges from what I told the individual. So no system level calibration and tweaking involved. Is that possible? I asked myself. And I thought probably it will always be difficult in nature, because in nature we can never fully control environmental conditions, but in the lab it should be possible. So we have an aquarium, we can control temperature, food resources, everything. We have this nice Daphnia. So this is the example that has been mentioned by Robert. This is from the Ben's PhD thesis. And without going into detail, yes, it was possible. Again, observed values are the dots. This is 40 days. This is the abundance of the Daphnia population. This is the total abundance. Daphnia babies, juveniles, adults. And this is the case for which the model was developed. The data were taken from individual growth and reproduction data. So no population level data were used except one parameter which was needed to make the model work. There was one weak spot in depth theory which needed to be fixed, but this is detail. But then the same model could be used to predict also the response of the population to high food levels, to other temperatures, and also, that was shown by Robert, the effect of toxicants. Yeah, so true emergence under controlled conditions is possible. And even when these figures were there, this is now the anecdote I like to tell very much. We wanted to submit this paper showing these results because I was so happy about this true emergence to American Naturalist, which is a high-ranked journal. But I was still quite worried. Will they really understand how cool this is? Maybe because of this long explanation of the starvation submodel, maybe they still might suspect that we still did some hidden tweaking or whatever, which we did, of course. But in principle, we did not. So Ben was there in the afternoon, and we were discussing how can we convince reviewers and editors how cool this is. And then I said, or do you know any other cool patterns with Daphnia, which is so striking? And if your model could reproduce it, in addition to this lab data, which we knew from the beginning, that would be really nice. And Ben said, yes, of course, there have been these 2 Nature papers where under certain conditions in the lab, Daphnia saw small cycles. Then they switched to large cycles, and after some time, they switched back to small cycles. Quite cool. I said, yeah, that would be really nice. I said, but don't invest more than 2 weeks in this because we want to submit it. 2 weeks would be, if it fails, it's good, but if it works, it's nice. That was at 5 o'clock. Next morning, at 9 o'clock, Ben came and showed me this figure. I said, what is this? I said, Ben, yeah, you told me. Small cycles, large cycles, small cycles. I said, did you stay awake all night? What did you do? Did you tweak 100 parameters? No, I just had to find the right level of resources, and it was just there. And so this is a kind of independent prediction and validation which you do not get very often in ecology. Therefore, I'm so happy about it. This is my feeling. But anyway, emergence from what? From first principles. First principles is always with quotation mark because we are not going down to the quarks. But in the field of ecology, it would be energy budget. We have talked a lot about dynamic energy budget theory. There are also other energy budget approaches, as Robert mentioned. Fitness seeking is an important principle. You can model it explicitly. The Trout model of Steve Welsbeck is modeling fitness seeking explicitly, but you can also assume that a certain behavior is improving fitness, so implicit fitness seeking. Then, of course, population dynamics is driven by variation of resources, all kinds of resources in space and time. For aquatic organisms, sometimes stoichiometry has proven to be very important first principle. And here, maybe we add to this list. So far, my list went up to here. So now maybe we add subcellular mechanisms. So, Valerie's and Richard's dream. Molecules, ecosystem service. Piece of cake, right? You do some magic trick, and then you get your 3 numbers characterizing perfectly ecosystem service. This is a dream. So, but what is the interface between molecules and ecosystem services? So where does it link? And I think the answer is obvious by now. It is the individuals. Because they follow the same program, except that guy here. Eat, survive, reproduce. So evolution is really the guiding principle here. Nothing makes sense in biology except in the light of evolution, a famous quote by Dobzhansky. And a less famous quote by Grimm is nothing makes sense in ecology except in the light of the individual, which is essentially saying the same, but in other words. So this is what I called, it's the individual stupid. I think, although all kinds of model types keep playing a role for all kinds of questions, individual-based approaches in this field for predictive systems model will play a special role because they are really linking processes which take place at the level of the organism, behavior, metabolism, energy budget, to system level properties. So, but now, it's hard enough to develop an individual-based model of populations, but what about communities and ecosystems? And here the advice is combine these 5 elements, maybe more, but these 5 I could think of, and then developing models even of communities and ecosystems becomes feasible. I already mentioned that you should distinguish very carefully between prediction and calibration. Have emergence from first principles in mind. Try to do this as much as you can, depending on your resources and time and data. Use multiple patterns to design your model, to test your model, to improve model design. And now, the remaining time, I will show a briefly example. Standardization of submodels. DEP is one example. There are others. And trait-based approaches using community assembly along environmental gradients. This is the approach which I think will play a big role in community ecology over the next years. So benefits of standards, they save time. You can reuse things. You use tested submodels. DEP has been tested in many, many cases. You don't have to debate all the time about all the equations because the properties are known. After sometimes you don't need to defend your submodel anymore. Okay, DEP, it's fine. I don't like it, but okay, carry on. Easier to communicate. You just say DEP, and everybody either runs away or says that's okay. Differences between species. You don't have a new model structure for every new species. You can use different sets of parameters, which make a systematic comparison easier. So it's easier to compare models of different systems. And what kinds of standards? It could be standard models of interaction. Plant people are quite good with this. They have zone of influence models, so circular zone of influence, or these gap models, which model vertical competition for light. These standard models of interaction have been proven extremely powerful. They're used in hundreds and hundreds of models. Standard models of behaviors, we are not there yet. But I think, why not standard foraging model, standard habitat model? I had a visitor working on tigers in Nepal, and he wanted to develop a home range model for his tiger. And I gave him a paper about the common shrew, where we had to develop, by Magnus Wang, a common shrew home range model. And he looked at the model and said, well, I just rename common shrew to tiger, then I'm there, because this is exactly what I need. So things like that, right? That in that case, the common shrew was trying to add pieces of the habitat to maintain a certain level of resources. That, what in most cases is important also for the female tigers. So simple principles, and then you can reuse model designs. Then, of course, standard models of life history, bioenergetic models, or the other proponents, metabolic scaling theory, so ontogenetic growth models, or things like that. So one example, a grassland community model, the purpose was to evaluate the response of this community to different grazing pressures and different resource levels in the soil. So how does species richness, productivity, and a diversity measure respond to different levels of grazing and resources? And the model, just briefly, distinguishes above and below ground competition. It has zones of influence. So the influence, you could think of the root system or the leaf system. They are separate, above and below ground. You have an empirical model describing growth. We have now a version which is based also on ontogenetic growth, based on metabolic scaling theory, but you could also use the DEP model here if you would prefer. If the zoes of neighboring plants overlap, we have competition. We distinguish between symmetric and asymmetric competition. We have resource levels, in this case, very simple, as constant parameters. We have adaptive resource allocation to the limiting compartment. If competition is stronger below ground, then more resources are invested in the below ground department. This is done in more sophisticated physiological plant models a lot. Traits, for example, seed production, seed weight, dispersal distance, establishment chances of seedlings have been specified, and then we have grazing, so there's biomass taken from the plant when they are grazed. There's one imposed factor. We had to assume that interspecific competition is stronger than interspecific competition. Otherwise, we could not reach the same levels of species richness which have been observed. So there we are hitting the boundaries of our mechanistic approaches. You just had to impose it. So this is the same here in figures, above, below, zone of influence, overlapping, size symmetric and size asymmetric competition. This is the number of state variables, the number of traits, and this number of traits then are used to define different plant functional types. For example, you have large plants which have large seeds, but they have small dispersal distances. You have small plants with small seeds with large dispersal distances. You have traits, trade-offs in your traits. So then data were taken from a database. So first it was a totally artificial model, then data were taken from a database. And to cut the story short, here's the output. Again, as I started with, is this a good match or not? Here we have extensive grazing, medium grazing, intensive grazing. This is the empirical side. This is the model prediction of simulated patterns. Here we have low resource levels. Here we have high resource level. And the output variable which we look at here is the species richness. And David Ford again would say this is really bad. I would say this is really good because there's no calibration involved at the system level. We took the values for the trait parameters from the databases. We assembled about 90 different plant functional types, let them invade in the system, and at the end about 20 survived. And the response of that system to different grazing and resource level was captured quite well. And the same story if you look at the Shannon index. Here we have this hump shade response qualitatively, which is quite good. The absolute levels differ to some degree. And here we have, for example, some divergence. Here's a trend, whereas here it is more or less the same. So it is not perfect, but again, this is without calibration, right? So you can see if you use first principles, in this case, it was a standard submodel of interaction, sigmoid growth model, and you use traits, which allows you to define functional types. Then you use assembly, put them together, let them interact, and just see what happens. You get relatively fast. I was really surprised by this. And completely independently, almost the same approach has been used here by Enrique and by Don DeAngelis. They modeled assembly of fish communities. I think they used also these principles, more or less. And also in that case, you can see that they could reproduce a wide range of observed patterns of real fish communities. Although at the beginning it might look quite artificial. So this is a very powerful approach. I hope you keep this in mind, that we have means, although IBMs are computationally costly and so on, but it works. So this is the advice to do, just a repetition. And before I conclude, here a little disclaimer. Of course, this is not true. I'm totally convinced. So types of models, of course, we need them all, but IBMs play a specific role, that's for sure. Be careful with the term prediction. It's really important to distinguish here carefully. Multiple patterns, in this context here, integrated systems models across scale is absolutely decisive. Emergence distinguishes from imposed properties. The more emergence you have, the higher your predictive capabilities. Individuals are, so to say, the interface in this business here. And it is possible, using all these things, to develop quite predictive and powerful models of communities, probably also ecosystems, but we haven't done this so much so far. Thank you very much.